Without further ado, we could probably move on to our keynote speaker. And I don't see, is this, okay, Dr. Jae Ryu. Just to give a little, so Dr. Jae Ryu, he's an associate professor of neurology with the Gladstone Institute of Neurologic Disease at the UCSF. Am I correct in that? Okay. I'll just invite you to come up to the podium to introduce yourself and to go through your very interesting discussion this morning. Thank you.
Good morning, everyone. My name is Jae. I am a scientific program leader at the Gladstone Institute, and I was assistant professor at the UCSF, but now it's promoted to the associate professor. Thank you. I'd like to thank the selection committee for giving this wonderful opportunity that I can share our research and the recent publication regarding blood clots and inflammation in COVID last year. So, at the beginning of the COVID pandemic, it was unclear why some patients infected with the strong COVID, severe COVID, suddenly developed massive blood clots. And clinicians observed that strokes, pulmonary embolism, microvascular clots detected throughout the body, including lungs and hearts, even in brain. So as the mechanism of the disease unfolded, it's become evident that SARS-CoV-2 infection triggered not only respiratory illness, but also systemic vascular and immunological dysregulation. And the coagulopathy and the neurological complication in COVID-19 still occur during the acute infections, and it persisted in long COVID patients. And even young COVID-19 patients, including those with mild and breakthrough infections. And previous studies show that the blood clot derived from the COVID-19 resist degradation despite anticoagulation treatments. Lately, long COVID has the features of the chronic vascular disorder that characterized by persistent thromboinflammation. In addition, hypercoagulability in COVID-19 is associated with the extensive fibrin depositions in inflamed lung and brain. The fibrin is derived from the soluble blood protein fibrinogen upon activation of coagulation cascade and forms the central structures of the blood clot. Others in our groups have been shown that fibrin depositions after BBB disruptions drive innate immune activation in several diseases, including autoimmune, inflammatory.
Excuse me for 1 second. I'm just going to bring your mics together. Okay, that's great. Thank you.
Yeah. And neurodegenerative diseases. So, the aspect that makes fibrinogen research important is the its unique structure-function relationship. What truly set the fibrinogen apart, among other blood protein is the coagulation factor. Soluble fibrinogen circulating in the bloodstreams. But when blood-brain barrier disrupted and soluble fibrinogen leak into brain parenchyma and convert into insoluble fibrin, persistent scaffold that deposit at the site of injury. And this conversion exposed the hidden inflammatory protein called cryptic epitope that binds to complementary receptor 3, also known as the CD11b CD18 receptor that expressed in several immune cells, including microglia, macrophage, natural killer cell, mast cell, etc. The other aspect of the mixed fibrinogen research interesting is there are genetic tools available, including fibrinogen knockout and fibrinogen mutant mice. Those tools allow us to study loss of function, to characterize when fibrinogen is deficient and the fibrinogen mutated, how they affect the neurological disease progression. So using those research, we and others have found that fibrin is both necessary and sufficient enough to induce neuroinflammation and neurodegeneration. What we have known so far, soluble fibrinogens leak into brain, become insoluble fibrin, and play dual role pro-inflammatory, pro-oxidant in innate immune cell microglia. Fibrin convert resting microglia into neurotoxic phenotype characterized with the inflammatory and oxidative stress gene signatures that are coupled with the coagulation factor expression, that promoting persistent fibrin deposit at the type of injury, and which lead to inhibition of repair, demyelinations, and neurodegeneration in several diseases, including multiple sclerosis, traumatic brain injury, and Alzheimer's disease. Recent study also show that the fibrin-linked neurovascular damage evident in COVID-19, and neurological symptoms and neuropathological alterations have been attributed to the secondary effect, systemic SARS-CoV-2 infection. Here, postmortem tissue from the COVID-19 patients markedly demonstrate the fibrin deposit, indication of the neurovascular injury. And at the site of the parenchymal fibrin depositions, we can able to see the reactive microglia can accumulated in the brain of the patient with COVID-19. Recent study using the enhanced MRI long COVID patient without brain fog, they found that BBB disruption strongly correlate with the brain fog in long COVID. And this subset of the patient also showed increased plasma fibrinogens that is can be considered as predictive biomarker of cognitive deficit after COVID-19 infection. However, the role of the blood clot in COVID-19 inflammation and the neurological change remain largely unknown. And therapeutic approaches to combat their effect are not readily available. So in this study, we investigate the fundamental role of fibrinogen in immune responses and neuropathology in COVID-19, and also explore fibrin immunotherapy-based approaches to reduce blood clot-mediated complication in this model. We took the multi-pronged approaches, including biochemistry and computational docking, and genetic loss of function study with infection-dependent, infection-independent. For the detailed signaling pathway, we use the transcriptomics and post-proteomic approaches. And lastly, we test the efficacy of the anti-fibrin immunotherapy against blood clot-mediated inflammation and neuropathology in COVID-19 model. we hypothesized that SARS-CoV-2 directly interact with the fibrinogen, forming blood clots and altering function, structure of these blood clots. So we created custom-based peptide array consisting of alpha, beta, gamma chains of fibrinogen and identified 3 key binding sites interact with the spike protein within the beta and gamma chain of fibrinogen. Those sites include the plasmin cleavage site in the middle, and the cryptic binding motif that is for the CD11B binding site, and an unknown region that might be involved in 3D conformational epitope exposure within the gamma chain, suggesting that multi-site interaction that might be crucial for in context of the blood clot formation and immune regulation. Using the computational docking, we identify the model spike region of the SARS-CoV-2 spike proteins and fibrinogen gamma region interacting site-specific manner. And using the alanine scanning mutagenesis, we identified the SARS-CoV-2 specifically interact with the C-terminus of fibrinogen gamma chains, suggesting that there are specific domain spike and the fibrinogen interacting each other, causing immune dysregulation in these cases. In this study, we have shown that the spike interaction with the fibrinogen in different animal model of COVID infections. And here we show the interaction between spike protein and fibrinogen by co-injection of the fluorescence conjugate spike S1 protein and fibrinogen in wild-type mice. And the 3D imaging of solvent-clear lung demonstrate that spike protein is interacting with the fibrinogen within the tissue, suggesting that these 2 proteins can interact during the infection. So we investigate that whether spike protein alter polymerizations, degradations, and the immunological aspect of the fibrin. When the human plasma was incubated with the spike protein in the presence of the thrombin, we observed very abnormal structure of the fibrin. And we also found that spike also delay plasmin-based fibrinolysis, indication of the abnormal fibrin structure formation. We also observed that spike increase fibrin-mediated reactive oxygen species production in macrophage. And we next studied the neuropathology and thromboinflammation in the animal model of the COVID-19 infections. In this study, we used fibrinogen knockout mice. These mice only deficient in the fibrinogen and other blood proteins still intact. And also we use fibrinogen mutant mice. These mice carry the mutant fibrinogen that lacking the CD11B, CD18 binding motif while retaining normal clotting functions. These mice infected with the SARS-CoV-2 beta variant, and we study brain pathology and lung pathology. In the fibrinogen deficient and the fibrinogen mutant mice, the macrophage activation, oxidative stress was significantly suppressed. And also inhibit the brain inflammations in fibrinogen deficient and mutant mice. We next study lung transcriptomics, and fibrinogen deficient mice suppress the pro-inflammatory responses and interferon signalings, and chemokine, and ROS productions. However, unexpectedly, we found that fibrinogen deficient mice enhance NK cell cytotoxic gene signatures, suggesting that the fibrin might have the regulatory function of the NK cell cytotoxic effect. To characterize how fibrin suppress NK cell function, we use bulk RNA sequencing method to characterize the specific gene signature by fibrin regulations. As you can see, NK-mediated immunity, cytokine, chemokine, ROS signatures, IL-2 signaling pathway, NK, NF-kappa B signaling pathways, all downregulated by fibrin in NK cell. And overall, the fibrin suppressed NK gene network, including mitochondrial functions, cytokine, chemokine production, and MET kinase pathway, and NF-kappa proliferation pathway was affected by the fibrin treatments. And furthermore, we used the post-proteomic approaches and kinase assay to show that the kinetics regulation of the protein, and we found that fibrin suppressed the JAK-STAT pathway, and the p38 MET kinase target was suppressed by fibrin stimulations. So we next investigate that what is the in vivo aspect of NK cell after SARS-CoV-2 infection. In fibrinogen deficient mice and fibrinogen mutant mice, as you can see, NKp46, which is the NK cell marker, is highly upregulated in this both mouse line, together with the granzyme expressions, along with the suppression of the spike protein in the lungs. That indicating fibrin suppressed NK cell and viral clearance after SARS-CoV-2 infection in these mice. So we wanted to explore the harmful effect of the fibrinogen in SARS-CoV-2 infection involved NK cell suppression effect. So the mice were infected with SARS-CoV-2 beta variant after depletion of the NK cell using anti-NK1.1 neutralizing antibody. And in the fibrinogen knockout, fibrinogen mutant mice, as you can see from the macrophage marker and spike protein marker, beneficial effect of the fibrinogen deficiency and the mutant mice we observe is completely rescued by NK cell depletion effect. Macrophage activations and spike protein detection level is reversed back to the wild type level. This indicating that the fibrinogen is required for SARS-CoV-2 infection, and the lungs and pulmonary lesion formation through the inflammatory activations and suppression of the viral clearance involving NK cell. So the persistent SARS-CoV-2 protein has been detected in COVID patients and long COVID patients. So we wanted to explore that spike protein and fibrinogen interaction can trigger thromboinflammation beyond active infection. So we used HIV virion pseudotype with the spike protein. And then we found that this pseudotype virion increased fibrinogen-mediated reactive oxygen species generation in the macrophage. And we further investigate the in vivo efficacy of the spike virion injection. After administration of pseudotype virion, we noticed that the injected mice developed a huge fibrin deposition in the lung. And when we compare with the fibrinogen deficient and fibrinogen mutant mice, we found that reduced macrophage activation and oxidative stress in the lung after pseudotype virion administration. So this study actually indicating that fibrinogen is necessary for macrophage recruitment, ROS production in infection-independent spike inflammatory responses. Our lab developed the anti-fibrin antibody called 5E8 against cryptic epitope expressed in the fibrin. These epitopes only expose the pathogenic fibrin, not the soluble fibrinogen. This antibody selectively targets inflammatory function without reverse anticoagulant effect. The fibrin-targeting immunotherapy has been tested in 2 different model systems, including multiple sclerosis and Alzheimer's disease. And we found that therapeutic strategy using the fibrin targeting suppressed clinical symptoms of the MS and increased neuronal survival and decreased immune responses in Alzheimer's disease model. And this anti-fibrin antibody is humanized, now called THN391, and has completed phase 1 trial, and is now moved on to currently tested in phase 1B in the patient with Alzheimer's disease and diabetic retinopathy. So we explore clinical efficacy of the 5E8 antibody against SARS-CoV-2 infection. We use the SARS-CoV-2 beta variant and Delta variant. A 5E8 treatment, as you can see from histopathological analysis of the lungs tissue from beta variant infected mice, 5E8 significantly suppressed macrophage recruitments, ROS, oxidative stress, and also decreased spike expression in the lungs. And unexpectedly, we also found that the NK cell granzyme expression is upregulated in 5E8 treated infected mice. And Delta infected mice, 5E8 also has effects suppressing microglia activations and also increase in neuronal survival. When we look at the transcriptomic analysis on the brain tissue, we found that antibody treatments suppress type 1 interferon signaling pathway and coagulation gene signature and chemotoxic response, and also reverse the homeostatic dysfunctions of the microglia after antibody treatments. In both beta variants and Delta variant infection can be considered as a non-neuroinvasive and neuroinvasive strain, because in Delta variant is acting on human ACE2 expressing mice, you can actually appreciate very invasive CNS penetrations. We found that huge fibrin deposit in both non-neuroinvasive and neuroinvasive animal model of COVID infections. And the fibrin deposit strongly associated with the microglia activation in the tissue. And these activating microglia also associated with the disease-associated microglia markers, including TREM2 and CST7 SPP1 messenger RNA in the brain of neuroinvasive COVID-19. We next tested the efficacy of the 5E8 in non-neuroinvasive beta variant infected mice model. We used prophylactics and therapeutic approaches. And the 5E8 treatment suppressed the microglia activation in the brain in both prophylactic therapeutic treatments of the mice. And these are consistent with the fibrinogen deficient and fibrinogen mutant mice. In both cases, microglia was hugely suppressed. So 5E8, the fibrin-targeting immunotherapy is specific to the cryptic domain epitope mediated by the exposed fibrin. In the Delta variant infected considered as a neuroinvasive model of the COVID infections, fibrin targeting using 5E8 treatments significantly suppressed the microglia activations and also increased neuronal survival in the brains. When we look at the transcriptomics regulation by the 5E8, here, all the significantly downregulated pathway by anti-fibrin treatments is IL-6 productions, chemotaxis, type 1 interferon productions, and response to interferons, alpha and beta productions pathway. So suggesting that overall, the fibrin-targeting immunotherapy suppressed brain inflammation in neuroinvasive COVID-19 model. In summary, coagulopathy in COVID-19 is not just a consequence of inflammation, but rather serve the apical driver of infection-induced thromboinflammation and neuropathology. In this study, we have shown that the fibrin promote neuropathological alterations either indirectly by inducing hyperinflammation through the modulation of the NK cell and macrophage in infected lung, or directly act on the microglia because it's fibrin's parenchymal deposition in the brain. Our data also emphasis of the enigmatic coagulopathy found in COVID-19 and showing that is causal role of the fibrinogen in the thromboinflammation in context of the COVID-19 infection. Lastly, our work suggests that the fibrin-targeting immunotherapy may represent new approaches to selectively suppress COVID-19 pathogenesis in the brain, other organ, without any adverse effect on normal hemostatic function of the fibrinogen. So this work is truly collaboration work from 5 academic institutes: UCSF, Stanford, the San Diego, UCLA, Glasgow institutes. Over 4 years of collaboration involved more than 50 scientists. We start this work at the beginning of the pandemic and then finish at the end of endemic. So it took 4 years to validate it and working together day and night, producing meaningful study so that it's beneficial to the patients and scientific community. And lastly, I'd like to thank the Carolina Akasoglu and Warner Greens. They are principal investigator from the Glasgow Institute. These 2 senior scientists contribute amazing work in the studies, bring all these scientists together, and then we can able to work together over the long years of the collaborations. In the pictures, we have 4 individual scientists, we equally contributing to this work. And then these are all the funding resources supporting our work over the years. Thank you so much.
Dr. Ryu, that was, wow, that was an amazing talk on the subject. Your method that you went through this to, and then get to this hope and promise of ways to treat neuroinflammation by targeting the fibrin antibody related. So that's truly an amazing talk. So thank you very much. We're going to open we'll open the floor for questions. For people who are attending virtually, if you have questions, can you connect to the Q&A section, post your questions there, and as well, if people have questions here in the audience, there's a mic there that we could, and what we'll do is we'll go back and forth between virtual and in person. So you have the floor for the first question.
Thank you for this talk. Really interesting, a lot of food for thought. The most common hereditary coagulopathy in Canada, over 1 in 20, is factor V Leiden. That affects the fibrinogen part of the clotting cascade. Are they more likely to have long COVID? And conversely, people with a factor I deficiency, which is again fibrinogen, a lack of fibrinogen, are they less likely to have long COVID based on your findings?
Wow, that's very interesting questions. Based on my knowledge, I don't know there's a large clinical trial actually indicating any correlation between the factor deficiency and the long COVID developments. However, based on our finding, the coagulation cascade actually promote the abnormal fibrin formation under the COVID infections. So I believe there might be relatable cascade events can be formed. However, we need to study large cohort of the patients showing if there's any correlation between this factor deficiency and the COVID development.
Thank you. And also you talked about NK cells. There seems to be some evidence that SARS-CoV-2 inhibits the NK cells at the very beginning of an infection. Does that have any effect on this? And what happens if you supplement NK cells or you plasmapherese and skim off the NK cells? Does that make any difference with your findings?
So yes, there are the clinical evidence showing that NK cell function is suppressed in SARS-CoV-2 infected patients. And so when the patients received more fresh plasma from the recovered patients, and they actually had a more strong efficacy suppressing the ongoing immune responses, I think might be due to the NK cell activity in these cases. Because of the other factors contributing to the NK cell suppressing effect, if we only focus on the NK cell activity, their efficacy may not be optimized. So in this study, actually, we wanted to emphasize that there are hidden role of the fibrinogen suppressing NK cell during the COVID infections. So it might be better to consider how we can regulate the inflammatory aspect of the fibrin so then we can boost NK activity together with the other reagent supporting the elimination of the SARS-CoV-2 viruses.
Thank you.
Thank you. Susan, just to ask, is there any online questions?
Good morning, everyone here and everyone in the foggy cybersphere. I'm Susan Law from Long COVID Web, and I'll be doing my best to try and capture some of the key questions that are coming in from the virtual attendees. But the first one is just, what do we know about the possibility of the COVID virus itself penetrating the blood-brain barrier? And does this account for the numerous symptoms?
So that's a very important question. We haven't seen any strong, clear evidence SARS-CoV-2 virus crossed a blood-brain barrier without any disruption in these cases. However, our follow-up study indicate that SARS-CoV-2 virus act on the vascular endothelial structures instead. So we believe that instead of the directly crossed BBB and replication in the brain, they rather act on the barrier and increase the permeabilize and the losing of the tight junction protein. I think that might be a more reliable scenario we can actually investigate it.
Okay, thank you. You have the floor.
Thank you for that talk. My name is Satish Rajam from Calgary. I have two sort of unrelated questions, but both to provide context for me anyway. So a few years ago, there was a lot of interest and talk in microclots, which, as I learned a bit more about it, is more fibrosis, which I think is what you're talking about, than as a cardiologist, what I consider a clot that you can give anticoagulants to. Is what you're talking about, is what you're finding the same as what the South African groups that described microclots found, or is it different?
You mean the microclot?
Yeah, the South African group sort of came up with an assay for it. It became all the rage for a while. I'm not sure if it has direct therapeutic implications or not, but is it the same thing and this is the therapeutic implication, or is it different?
No, I think that we believe yes, the microclots. Our study actually focused on the insoluble fibrin matrix protein deposit at the site of the injury. We're not implicating that the circulating any soluble fibrin ligands in the bloodstreams, which might think that might be considered as a microclots in the bloodstream. So we're not sure whether microclot actually expose the same cryptic epitope our immunotherapy can targeting at the same way. So even if they are coming out of the fibrinogen coagulation cascade, I believe that there's different property of the fibrinogen.
And the other question is, you had very nice slides, sort of cartoons where you evoke the spike protein as being causative. And my question is, did you look at other viral illness serum, like patients with other viral illnesses, right? There are a lot of patients with phenotypes that look like what we're calling long COVID now, that pre-existed well before what we called COVID, although who knows, that could have been, SARS infection too, we just didn't have a name. But the question is the spike protein critical or not? And part of why I'm wondering that is that your drug in phase 1 studies, you've been looking at in disorders far-ranging, and I'm wondering how much this has to do with spike protein COVID specifically, and how much this might be a broader issue for brain distress in a series of viral-bound illnesses.
So the fibrin immunotherapy, as you see, that not targeting the spike protein, is targeting specifically cryptic epitopes exposed in the fibrin structure. But based on our research, we noticed that spike protein very specifically engaged with the cryptic epitope of the fibrin. So you can see if antibody, because of the spike and interacting with the fibrin, potentiated fibrin-mediated inflammations in other effect. If we can interfere, these two interactions can suppress a infection independence, the fibrin-mediated inflammation, for example, in COVID settings. So I think that might be a little different aspect I can actually emphasize.
Okay, thank you. Susan, do you have any questions virtually?
Yes, there's another question. What do we know about mast cells, MCAS, and the role of antihistamines as potential treatments?
It's very interesting questions. However, this is not my the area that I can actually answer. But as far as I know from the literature, there's not much clear, reliable, large core of study the mast cell environments in the COVID patient symptoms. However, there are small clinical trials focusing on antihistamine suppressing mast cell or superactivations has mild effect in COVID-19 patient symptoms. Definitely, there are need for the large cohort trials understand these two connections for the progression of disease.
Okay, thank you. You have the podium.
Hi, I'll make it very quick. I'm Dr. Fumi Okanola. I'm a practicing family physician in Vancouver, Canada, and I'm also the founder of Long COVID The Answers, a website and podcast series. So I just want to clarify the process by which SARS-CoV-2 causes this stimulation of fibrin and fibrinogen that you've discovered, because unfortunately I haven't read your paper yet. And then I'm going to ask a question around that. So from my understanding of what you presented today, SARS-CoV-2 binds to an epitope on fibrin in our bodies and induces clotting and an inflammatory process. Am I correct?
Yeah.
Okay. And you've developed an antibody that can slow down or halt this process. You've identified an antibody that can slow down or halt this process and thereby reduce the risk of clotting. Am I correct?
Sorry, I didn't get the last question.
You've identified a form of treatment in the form of an antibody that can slow down or prevent the process of the binding of SARS-CoV-2 on the epitope of fibrin in our bodies. Is that correct?
So that's an important question. We demonstrate the efficacy of this antibody blocks spike interacting with the fibrinogen and drive the neuropathology and thromboinflammation in animal model. We haven't shown the antibody interfere these two molecules separated in vivo system. What we only show in vitro when antibody presents at the incubation of the spike protein and fibrinogen is completely interferes binding these proteins together. So we believe there might be similar efficacy in the in vivo system blocking spike interacting with the fibrin.
Right, okay. So my question is, bearing in mind, so there's an implication because we know that long haulers suffer from brain fog and that this whole process might be going on in the brains of long haulers, 80% of whom suffer from brain fog might be a contributing factor to it. How do you hope, I mean, and you suggested that we may well be able to develop some form of immunotherapy to halt this process. Bearing in mind that we haven't fully classified the different phenotypes of long COVID amongst the millions of sufferers that they are, how do you hope to identify who would benefit from this immunotherapy within this population?
I think it's solely based on the subset of the patient existing, showing the BBB disrupted in the long COVID patients and persistent fibrin deposit. There might be related to persistent immune dysregulation in the patient groups. and contributing to the neurological symptoms. In that specific cases, this antibody immunotherapy might interfere this ongoing the fibrin-mediated any inflammatory response related to the neurological symptoms. So I cannot say this immunotherapy might work every case of the long COVID patients, might be a subset of the patient who actually showing the BBB disruptions and persistent fibrin deposit might be, there's a potential, possibility this immunotherapy might work. But still, we are in the stage testing efficacy in other disease. So we might need to actually explore further more the clinical trials and the mechanistic studies down the road.
Yeah. And do you think, bearing in mind that fibrinogen, as you've said, is associated with the development of diseases such as multiple sclerosis and Alzheimer's, and I can't remember all the others, are we looking at, do you think that long haulers have a greater predisposition to some of these diseases than the general population? And are we looking at an increase in these diseases in the general population, bearing in mind the coagulopathic factor of SARS-CoV-2, do you think?
So I believe are you talking about the immunotherapy related?
Well, it's just that you identified that, fibrinogen is causing inflammation, causing coagulopathy, and is associated with any Alzheimer's and multiple sclerosis. So are we looking at an increase in those diseases in the general population? And also, do you think long haulers will be more predisposed to developing those diseases in the general population, bearing in mind
So
we're all being infected with SARS-CoV-2 persistently?
So we believe that the fibrin deposition is not just a specific disease. If the disease actually characterized with BBB disruptions, and there's a high chance our immunotherapy produce the protective effect against the fibrin deposit mediated histopathological change or neurological symptom related. So in not only neurological disorder known to be BBB disrupted, is the subset of the patient long COVID or acute infections. If there's any BBB disrupted, this immunotherapy or the fibrin mediated the neuropathological mechanism can be applied to this study we actually ever shown here.
Thank you.
All right, thank you. We have one last question. So the man in the blue jacket, you're welcome to ask your question. This will
Thank
be
you.
the
That
last
was
one for you, Dr. Yu. Thank you.
Thank you. This is a brilliant presentation and fascinating to me and my colleagues. We have fascinated by the fact that you used the trimeric spike protein as your inducer. And the rest question is in terms of looking at the potential ACE2 binding target of the spike protein. Is this same domain implied in your interaction? And was the activity somewhat based on the fact that you had the trimeric version to induce it? And the reason for this, of course, is that there's this potential theory that in long COVID, circulating spike protein still seems to be present in the absence of the virus. So it's that mystery, really, that may be inducing some of the symptoms, including the fibrinogenic activity in long COVID symptoms, that spike protein is present and continues to induce this. So in your structural determination, did you identify any different domains from the ACE2 binding domain?
That's very good questions. When we work with the trimeric, yes, the S1 protein, trimeric structure of the spike protein, we haven't looked at the specific binding site differentiated between these different types of the spike protein bind to where in the fibrinogen site. There's something we like to pursue in the future studies. But at this moment, we don't have actually the answer for your questions.
Great. Well, look forward to working with you on perhaps working that out. Thank you.
Good. Thank you for all your great questions. And thank Dr. Yu. It was a brilliant talk. And thank you very much for coming.
Thank you.
Thank you.